Okay, yeah, great. And thanks so much for giving me this opportunity to talk about our work to this group. So I'm Aryeh Warmflash. I'm, as it says, in the Department of Biosciences at Rice. I'm a physicist by training, actually, but I've gone into sort of stem cell biology. And my lab is very interested in studying early human development and what happens in normal human development. And we've also more recently, together with Project AP, got interested in whether we can use the systems that we've developed to study human development to also study disorders associated with human development. And the approach that we take is beginning with human pluripotent stem cells, which I'll get into what they are if you're not familiar, but they're cells that can basically make any cell type in the body. We try to recreate the process of normal development or aspects of it in a culture dish, and then use those systems as proxies for studying developmental events that otherwise wouldn't be accessible to researchers. And so like Ethan and alluded to, this talk is going to be a little bit more on the sort of the basic science side than maybe some of the other talks. So as they said, please feel free. I've tried to include enough background so everyone can understand it. But if something is unclear, please feel free to jump in and stop me. Okay, so I'm going to do this in 3 parts. So I'm going to give some background on early human development that serves as the background for what we study. And then I'm going to talk about these micropattern stem cell models that we've designed to study an early process in development known as gastrulation. And then at the end, I'm going to tell you about some of our early results applying these systems both to patient-derived cell lines from AP patients as well as to genes that we can modify in the lab that exist on chromosome 8p. Okay, so starting with the human development. So this is a sort of series of snapshots from what's known as the Kyoto Human Embryo Collection, and you can sort of get a sense of the change in scale and pattern as human development proceeds. So starting from a single cell on day 1, the scale bar here is, I think, 5 millimeters. So even the largest objects here are quite tiny, smaller than a centimeter, and these are really, really tiny. And the embryo grows and is further patterned. And we're interested in some of the earliest events that happen, which are right in this window, around 15 to 17 days. And these are some of the most important events for establishing the basic plan of the body, as well as the basic lineages for the cells that will form the body. And it's also in this window and the window slightly later than this that sort of the most things can go wrong. So most, well, first of all, most cases of infertility are associated with very early development, and also most disorders there are disorders associated with later development, but a large chunk of them are associated with these processes in the first month of development. And so it's interesting to study both from a fundamental perspective of how the body plan gets established, and also from the perspective of this is very important knowledge if we are to understand issues of infertility and developmental disorders. So if I sort of in a cartoon way zoom in on what's happening in that early development, so the embryo, of course, starts as a single cell. The single cell will make copies of itself, which are shown in yellow here. These are times, but these refer to the mouse embryos. This taken from a review article mostly about the mouse embryo. These things would happen somewhat later in humans. So this period that takes 5 days in mouse would take about 12 days in human or so. And then these cells begin to specialize in a series of decisions so that the embryo divides into 3 different tissues. The epiblast, which is the most important for what I'm going to be talking about, because the epiblast is what goes on to form the entire embryo proper. And then these surrounding cells, which are called the trovectoderm, these are what will form the placenta. And then these primitive endoderm cells will form other extra embryonic tissues. So basically the entire embryo derives from this epiblastic tissue. And the important thing to realize from what I'm going to talk about is that the human pluripotent stem cells that we work with in the lab, they are the in vitro equivalent of these epiblast cells. So if you could imagine basically taking these epiblast cells out of the embryo and culturing them and then maintaining the properties that they have in the embryo to a large degree, that's what the cells that we have are. And that's important because they are the cells that during normal development would generate the entire embryo proper. So this sort of summarizes the decisions that these cells make. So you start with these single cells that I showed you. There's a series of decisions that separate the extra embryonic tissues like the trophoblast, which will give rise to the placenta, and this hypoblast that I showed you from the epiblast. And then the epiblast, the first thing it does is decide between the three germ layers. So the ectoderm will form the nervous system and the skin, and the endoderm will form the digestive tract and other organs, and the mesoderm will form things like muscle, bone, blood, as well as the heart. So the cells that we work with are here in the epiblast and are sort of poised on the verge of making these first decisions between the different lineages that exist in the embryo. Another important thing to realize is that while most of the cells that we work with actually derive from embryos, so you can think of them as taking the cells out of the embryo and culturing them, there are alternate routes to making cells that are essentially equivalent to those cells. And this was first shown a little more than 15 years ago now by Shinya Yamanaka's lab. And what they did was they found that if you take a normal cell from the body, so you could take skin biopsy from someone or blood from someone, and you transduce them with particular transcription factors, so particular genes, they can be reverted back to this state of pluripotency. So back to a state where they would, in theory, be capable of generating an entire organism. And that's been sort of rigorously proven for mouse because people have grown entire mice from these cells that have been reverted. For human, of course, that's not possible, but as far as we can tell, they're functionally equivalent. This is just sort of a little snapshot from this paper where Takahashi and Yamanaka are looking at how many stem cell colonies you get by adding different factors. And it's a cool paper where they started with basically everything they can think of. And then they start, they get stem cells, which is amazing. And then they start subtracting one factor at a time to see what are the minimum requirements for making stem cells. And in the end, they defined these 4 factors, which are now famous. And basically by taking almost any cell and throwing these 4 genes into them, that cell will be reverted to the pluripotent state. So Yamanaka won the Nobel Prize for this work remarkably quickly. It was published in 2006. He won the Nobel Prize in 2012, along with John Gurdon, who had shown similar things for frog in the 1950s. So it took him much more than 6 years to win the Nobel Prize. So coming back to the embryo itself, we are very interested in this process where these cells make these early decisions. And this is a process that is not just about cells making decisions, but that plays out in space and time. So this is sort of a schematic of that early embryo I showed you, where the whole embryo proper will be generated from this pink tissue here. And then over the next week, it's transformed into something that looks more like this, where the embryo proper starts out as this sort of single layer of cells, but eventually generates this 3-layered structure. So that's where the origin of this term germ layers comes from. So you'll have the layers of ectoderm, mesoderm, and endoderm, and the mesoderm and endoderm are generated from the ectoderm through a process in which they're generated in a localized region known as the primitive streak, and then they migrate away from this region and generate these additional layers. And so there's a lot going on during this time. The cells are changing their cell fates. They're also moving quite a bit. The whole embryo is being reshaped, and simultaneously the axes of the embryo, so where the head versus the other side is going to be, where the front versus the back is going to be, those are simultaneously defined with these processes. And I like to show this movie. Of course, nobody's seen this process in human. This is a frog embryo, but this is the equivalent stage of development in a frog embryo. The advantage of the frog embryo, of course, is that it just develops in what I've heard scientists refer to as basically pond water, and you can observe it happening, and it also happens very quickly. So whereas this process, the gastrulation stage lasts for a week in humans, it lasts for less than a day in frogs. And so this is a movie where you see the frog is basically making these additional layers through these cells involuting onto the inside, and now it's also repositioning the tissues to make the axis, so it's elongating so that the head will form over here in the top right and the tail will form over here. So you can get the sense that this is sort of a tremendously complex process. There's a lot happening. It's also a tremendously dynamic process, and we're interested in studying the process in humans, so that's a very difficult thing to do. And we've tried to take this reductionist approach of starting with the cells that will generate the embryo and working with those cells in trying to sort of trigger them into do events that are at least somewhat like what you're watching so that we can study them. It's known from many decades of work in things like the frog, but also in mammalian embryos like the mouse, that there's a set of conserved pathways that are very important for these processes. And in particular, there's a signaling pathway known as BMP, which stands for bone morphogenic protein, which derives from this blue extraembryonic tissue. It triggers a second pathway, which then triggers a third pathway, which operate in the embryo itself, and these are known as Wnt and Nodal. And somehow these pathways are important for allocating these different cell fates and triggering these different movements that we saw. But we really don't understand very well, especially in mammals and especially in human, how these signals get organized in space and time in order to trigger this process, and how the cells of the embryo interpret these signals in order to make decisions between the different fates they're going to become and the different movements that they can make. And there are very limited options, especially in human, for studying these questions. I just want to sort of briefly mention, so I started talking about signaling. I'm going to talk a lot about signaling, and this is what I mean by signaling. So if you can think of a cell, the cell has a nucleus where all of its sort of decisions get made, where its DNA lives, and then the rest of the cell is called the cytoplasm. And signaling means that there are extracellular molecules, which we call morphogens or signaling molecules, which are secreted by cells and then received on the surface of other cells with receptors. And these trigger processes inside the cell, which eventually end up in the nucleus and changing the genes that the cell makes and ultimately changing its cell fate. So when I refer to signaling and cell fate decisions, it usually takes place within a hierarchy like this, where there's some proteins outside the cell that trigger signaling cascades inside the cell, which make changes to the genes the cells are making in the nucleus and thereby affect the fate of the cell. And so it's these processes that we're interested in and how they're organized in space and time. And sort of the classic question in developmental biology is, what is the relationship between these signaling molecules outside the cell and the fate that the cell becomes? And sort of the classic answer is that it probably has to do with how much of these signaling molecules that you have, right? So in this model, which was proposed a long time ago by a famous developmental biologist, Lewis Welpert, so if you have a lot of a particular signaling model, you make one type of cell, which is shown in blue here. And if you have a medium amount, you make these white cells, and less, you make these red. And he called this the French flag model because it generates these striped patterns similar to what you would see on the French flag. But there's very little actual evidence for this model, and I hope to show you that for at least the systems that we work with, which we think model human development, this model is actually not correct, and something much more dynamic and complicated is happening. Okay, so I'm going to tell you that that's sort of by way of background. If there's any questions, feel free to sort of chime in now, but I'm going to segue into telling you about these micro pattern models that we've created to study these processes that happen in the embryo, but to study them in vitro and to study them with human cells. So when I started working on this some time ago, we had the sort of simple idea that, we have these cells. They're cells that grow in vitro. They're the equivalent of these epiblast cells that make the embryo proper. It's known what the signals are that are involved. So maybe we could just sort of dump these signals on these cells, and this will trigger similar processes, and that will sort of allow us to study the process in vitro. The problem with that turned out to be that when you do that, the cells in fact do differentiate, and they make a lot of the fates that you would see in vivo, right? So here I'm getting ectoderm in purple and mesoderm in yellow and extraembryonic cells in blue, but it's making a big mess out of this, right? So the cells' fates are sort of all jumbled up, and I'm not getting coherent territories of fates. And so we started to think about, well, what's the difference between this culture and what happens in the embryo? And the obvious difference was that in a standard culture, you don't control the geometry in which the cells grow, whereas an embryo always has the same size and shape every time. And so we started to grow our cells like this instead, which takes advantage of a technique called micropatterning, which basically means I chemically pattern the surface so that the cells can only stick in particular places. And so that allows me to make perfectly circular, for example, colonies where the cells are confined to the colony. And when I do the exact same experiment I did here, but I do it on this confined circular colony, I now basically remove the variability associated with the geometry, and you can see that I make very nice patterns of different cell fates in a very particular order, which in fact corresponds to the order of these cell fates that you would see in the embryo. And so, we've been excited about these systems for a long time because they're systems that enable us to sort of study this process of embryonic patterning in vitro. And what I want to emphasize is that this process is almost completely self-organized. So we're not forcing any particular cell to have any particular fate. We're dumping it, we're growing the cells in these circles, we're treating them with a single protein, a single growth factor, and downstream of that growth factor, they organize themselves into these patterns. And so the sort of central question that's been driving our research is, how do they do this, right? What is the mechanism by which these patterns are made, which we think is likely the same mechanism by which patterns are made in the embryo in vivo. This just gives you some sense of how different these cultures are from regular cultures. In regular cultures, you have all kinds of sizes and shapes, and all kinds of funny things happen that lead to the mess you saw on the previous slide. And then we can generate many very reproducible colonies of the exact same size and shape. We can make them in different sizes and shapes to see the effect of size and shape and so on. And so this is a system we've been working with for some time to try to understand these. And
Aryeh, if you could just pause for a second, if we go back,
sure.
Just want to make sure, because a lot of people may be feeling shy or whatever, but
yeah,
they want to understand what's going on, right? I mean, maybe you look at this and you're like, what's up with these crop circles? Like, if anyone has any doubt in their mind of what is going on here, right? Obviously, there's a surface here. We're zooming in. You're seeing that the cells are only allowed to grow on, and you can't see what you've done to the surface there. And I don't want to go into all the chemistry, right? But I just want to pause here. If anyone has any doubt what's going on here, please raise your hand, because this is going to be key, I think, for everything going forward. But if no one has any questions, then we can go on, or just use the chat if you're feeling shy to speak up. But I just want to give people a chance to just digest that and then ask any questions. All right, Aryeh, sorry for the interruption. Please continue.
Oh, that's fine. Yes, and I agree. Please jump in. And maybe one thing I should emphasize is that each one of these circles, is sort of, this picture is sort of zoomed out. So each one of these is patterned in this way, right? So if we were to look at the cell fates in this culture, you would get a series of these sort of bullseye patterns in every single one of these circles, which, you can almost think of each one of these as a separate sort of proxy embryo that allows us to accumulate statistics on what's happening in these colonies. And that's in contrast to here, where every colony sort of has its own individual personality, and it's hard to say, how will this colony be different from this colony? Here we ensure that they're all the same. So the first question we had was, is it that
Faye, go ahead.
Sorry, I just thought I would take the opportunity, Ethan, just to make sure that I do understand correctly.
Yeah,
so where you've got those different size circles, Aryeh, is it that you've basically chemically patterned them in different sizes, right? So they're different size bullseyes,
yes.
Is that it? And each one is a separate culture?
Yeah, so they're not really separate cultures. In brief, the way we do it is that we take a molecule that cells and protein won't stick to, and we cover the whole surface with it. And then we take a mask, block UV light through the mask, and it burns off the pattern that you see. So that stuff that cells don't like is gone, and the protein won't stick to is gone. And then we'll take the surface and we'll coat it with something cells like to stick to, so extracellular matrix proteins. And so what you end up with is complementary patterns of these extracellular matrix proteins that cells like, and these other molecules, these sort of cell-phobic molecules that cells don't like. And so when you seed cell, you only have one culture, but when you seed cells in that culture, they stick in these very particular patterns. And then each one of these little circles, because they can't leave the circle, right, the surface in between here has been passivated. these things are basically independent, right? So we've looked at this more quantitatively, and you see no correlation. This colony is not mathematically more likely to be similar to this colony than to a colony over here. So each one is sort of its own independent object.
Great. And the extracellular matrix you've laid down is the same for each circle. So like in the middle, like, you've got your three zones, right, in each circle, but the extracellular matrix they're sitting on is the same for all of them.
Yeah, exactly. Because we just basically burned off the other stuff in this pattern, and then we washed the matrix proteins over it, and it just sticks everywhere we burned it off. So it's all done with light and chemistry. And there's no specific deposition of anything in any one place. It's just the mask that we have that yields that sort of pattern.
It's fabulous.
Okay, anyone else want clarification? Don't be shy. Otherwise, thanks, Faye, for leading the way there. All right, let's onward.
Okay, so the first question we tried to address was the sort of, is this model where you have different levels of a signaling factor, does that make sense for these cultures? And so the idea would be that, we're treating with this protein called bone morphogenic protein, and maybe somehow the cells at the outside see the highest level and the cells at the center see the lowest level. And so to test that, what we did is we basically used the same technique, but shrunk the colonies down until they're very tiny, so that every cell would be like a cell on the outside. They're not different, you don't get patterns, but then we can vary the level of the protein and ask, can we cycle through these different cell fates? And the answer is no, you just get a switch between two cell fates. So the cells that are stem cells are green here, and the cells that are they become like the edge cells in the previous picture, so they're extra embryonic, they're red here. And what you see is that as I elevate my concentration, every cell that I see is basically green or red, but no cells become anything else. So these intermediate fates, which make circles in the middle of the colony, I never get those at all in these small colonies. And so I cannot replicate the patterning of the big colony by controlling conditions very finely and changing the concentration. This is a quantification of that, which is basically the stem cells are quantified in blue and the extraembryonic cells in red, and the sum of them is in pink. And basically the sum of these two cell types accounts for more than 90% of the colony, no matter what dose of this protein I use. And so we can't, and the green quantifies the cells that have one of these intermediate fates, so we never see it. And that sort of disproves this model, but if you're a serious developmental biologist, that's probably not very surprising to you, because it's known, as I mentioned earlier, that there's a cascade of signals involved in this process. So the BMP signaling that we're adding needs to trigger Wnt signaling, which needs to trigger nodal signaling. And so BMP alone, it wouldn't really make sense for it to be responsible for these different cell fates. And so we tested if these Wnt and nodal signals are responsible for these fates like they are in the embryo. And in fact, they are. So you can use small molecule inhibitors to block signaling through these pathways. And if I block signaling through Wnt or signaling through nodal, I basically lose these intermediate factors. And so then the colony is partitioned into these two fates, the cells that responded to BMP at the edge and the cells that didn't at the center, but my intermediate layers are lost. And so again, to emphasize, we are not adding, in this condition where you make these, we are not adding any Wnt or any nodal. The cells downstream of the BMP signals that we are adding make their own Wnt and nodal, and they make them in particular patterns, and that's part of the process by which the cells self-organize to make these colonies. And so we're excited about this because this means the cells are making their own signals, they're communicating with each other, and we can study the process by which the cells communicate with each other in order to make these patterns. And so the approach that we've taken to do that is that we wanted to use fluorescent proteins so that we could make cells light up in a particular way when they receive these signals. And to understand how this works, I won't get into all the details, but basically what you need to understand is that when cells see the signal on the outside of the cell, they send particular molecules to the nucleus of the cell. So in this case, I've highlighted SMAD4. So when they see the signal through BMP or in fact through nodal, both of these pathways use SMAD4 and it goes into the nucleus. And so we can use CRISPR technologies, which are basically technologies to edit the genome in any way we would like, to modify the SMAD4 locus in such a way that we've stuck a green fluorescent protein on SMAD4. And for the Wnt pathway, there's a protein that plays a similar role known as beta-catenin, which moves into the cell nucleus. And we made a different cell line where we modified those cells where beta-catenin is fused with GFP. And so now we can watch, we don't, in a non-destructive way, using the microscope, we can watch in space and time and see how these cells are receiving these signals.
And I guess those papers show that, what is that? I guess like N-terminal fusion of GFP doesn't mess up the native function of SMAD4 or beta-catenin. Like that was all, they did all those controls in these papers.
Yes. I mean, that's very. Compare the signaling response in these cells to regular cells, compare the differentiation, and show that everything is basically the same. And that's not a given, right? We've, some of these things we've made, like for example, if you put this fusion on the C-terminus, you'd trash the protein. So it really matters how you make these things. But yes, those controls are in these papers. So these are kind of examples of the kinds of movies you can get with these and the kind of data we can quantify from them. So first for beta-catenin, so this is the Wnt response. So beta-catenin is also a component of what are known as adherence junctions, which are junctions between cells. So these lines you see are borders between two cells. But then when we add Wnt protein, you'll see this beta-catenin accumulate in the centers of the cells as well, including in the cell nuclei. And we can quantify, we have a separate marker for the cell nucleus. We can quantify how much of this is in the nucleus as a function of time at different doses. And you see this. And similarly, this is the SMAD4 reporter. So these holes now are the nucleus. It's all over the cytoplasm, but excluded from the nucleus. And then when we add the ligand, you can see the nuclei fill in. And each of these pathways has its own characteristic dynamics, which was a little bit surprising to us. A lot of people think of these pathways as sort of switches where they're, outside the nucleus, then you add the ligand and they go in the nucleus. But actually, after you add the ligand, they each have their own dynamic properties, right? So, for example, this activin pathway, even though the ligand stays in the media and is active the whole time, the cells become desensitized to it and basically shut off their response. And it doesn't matter how much of the ligand you have. Whereas for beta-catenin,
oh, sorry, I was gonna say above a certain dose, though, you never go back to the baseline of.
Yes, that is true. They're almost completely what we call adaptive, but not completely adaptive. And this baseline may be important for the expression of particular genes. That is true. And for beta-catenin, you sort of see the same thing, but it's more dramatic and dose responsive, right? So these adapt to beta-catenin, but that adaptation is essentially full at lower doses and very partial at higher doses. And actually for BMP, which I'm not showing, they don't adapt at all. So every pathway has its own sort of characteristic response dynamics, which we think are important for understanding how the system is working. So what's really interesting is to then go into this system and to measure the signaling dynamics as these colonies are getting patterned. And so I'm going to sort of summarize a lot of work in a few slides. But the first thing that happens in these colonies is that the cells all respond to the ligand that we create, but then the response gets pushed to the edge of the colony. So here you can see this is just a snapshot at 40 hours, where you can see this protein accumulated in the nucleus of cells. Only at the edge and not signaling at the center. And these are precisely so the cells that are high for this protein are precisely the cells that adopt an extraembryonic fate. And this makes sense with what I showed you before, with this sort of switch between pluripotent or stem cells and extraembryonic cells, because it's this pathway that governs that switch. And so what we learned from this is that what's actually important in this decision is not the amount of this protein, the amount of the signal you have, but the duration in which it's in. So it's prolonged at the edge and transient in the center. And the ones with prolonged signaling are the ones that adopt this extraembryonic fate, but that this pathway doesn't explain the other circles that you get. So to explain that, you need to look at these other two pathways, Wnt and Nodal. So this is a movie of what this Wnt reporter looks like in time. And now remember that when you see these cell junctions, these are sort of the adherence junctions. These are not active signaling, but when you see the cells fill in, that's when the signaling is active. So you see sort of a bunch of cell movement, but not much signaling here. And then you'll see the signaling become high sort of in this region here. And then that region sort of starts to spread towards the center. So that's quantified here, where time is this axis and space is this axis, and more signal is more red and less signal is blue. And so what you see is that, right, the signaling sort of initiates at a particular time. It actually takes a while. It takes about a day before the signal initiates. It initiates in a particular location, a certain distance from the edge, which is about 100 microns. And then this territory sort of spreads. This one signal crucial for making these layers. And as the signal spreads, it triggers this second signal further down the cascade, which is known as nodal. I'm not showing you the movie here, but this is the similar quantification of nodal. So it starts a little bit later at 30 hours and then spreads in. It actually spreads faster. So the wave of Wnt triggers this wave of nodal, but then the wave of nodal actually nodal moves on its own. And if you measure where you're getting differentiation to one of these inner layers, so you can measure that in time, and then you can compare that with this signal, and that's summarized in this plot here. So the signaling through nodal is shown by two different measures in the green and the purple. The purple is the live cell imaging, so we have quite fine quantification. This is sort of controls where we fix cells, but the timing of differentiation is shown by these shaded cells. So it's exactly in the wake of this second expanding front of signaling that triggers cells to differentiate. And interestingly, I don't really have time to talk about this, but these waves of signaling will actually keep going. So they go further than the territories of cell fate that they mark. And what we think is actually important to the cells, like what are the cells reading as these waves go by that make them adopt particular fates, is the relative time difference between the different waves that go through. Because so the first wave gets triggered earlier but moves slower. The second wave gets triggered later but moves faster. So that means that the second wave is actually catching up to the first wave and eventually overtakes it. So every position in the colony sees a different difference between those waves, right? The closer you get to the center, the more likely you are to have seen the second wave before the first wave. And we think that that interval is sort of what instructs cells, at least for some of these fates, to become those fates. Okay, so I'm just going to tell you a tiny bit more about this system. This is sort of the latest stuff that we've published that we're excited about, is that everything I told you about is basically looking at the response of cells to these secreted signals, but not looking at the secreted signals themselves. And that's something that in the field has been tremendously difficult to do, because these secreted signals, they float outside the cells, they're present at very low concentrations, they're very hard to study. But we thought that in these cultures, which are flat and in 2 dimensions and ideal for microscopy, maybe we could visualize these secreted signals. And so we started by looking at this most downstream pathway, mostly because there's this nodal pathway is the nodal for this pathway is the only protein that's important for this pathway. If you knock out nodal, you completely lose signaling through this pathway at this stage. And so this allows us to study this one very important ligand. So a postdoc in the lab through sort of complicated things that I don't have time to talk about, but was able to make a fusion of a yellow fluorescent protein with nodal that we were able to show in this paper was fully functional. And so we can watch exactly where the cells make nodal and how that gets around. So this is a similar movie. It takes, as I mentioned, it takes about 30 hours for nodal signaling to start, and you can actually see the production and spread of the nodal protein. And that line enabled us to do experiments like this. So I should have mentioned more clearly, Lefty is an extracellular inhibitor of nodal. So there are proteins that the cells make and secrete that bind to nodal and prevent it from working. And so here we have fluorescent cells that make fluorescent nodal, and where Lefty is marked. And then on the other side, we've juxtaposed them with cells that don't make fluorescent nodal. And so we can ask, how far can this fluorescently labeled nodal spread from the source that make it into the territory of cells where they make nodal, they just don't make this fluorescently tagged nodal. They're their cells are wild type in the nodal locus? And the answer is it's hard to see, but they only spread exactly onto the cells that touch them. So this was a very surprising result, because this is thought as an extracellular signal that gets secreted and diffuses and can make it to cells sort of several cell diameters away. But we only saw it immediately at the junction. Lefty, in contrast, the inhibitor can actually spread quite far. So you see it, 6 to 8 cell diameters away from these cells that are producing it. And to prove that that's really a functional thing, so nodal can only spread to the neighboring cells, what we did is we knocked out nodal in some cells. So the cells on this side now either can make nodal if they're wild type or can't make nodal if they're nodal knockout. And we asked whether the signal, as represented by this protein moving through the nucleus, can spread. And so if these cells can make their own nodal, the signal can spread all the way through, right? So these cells were induced to make nodal, and then the signal spreads, and you can see it in the nuclei of all these cells. But in this case, these cells were induced to make nodal, and it goes to the nucleus. But since these cells can't make their own nodal, only the cells immediately at the junction signal. So what this tells us is that the way the nodal protein is working is actually through a kind of relay game of telephone. So each cell, when it's induced to make nodal, can make nodal. It can then pass that nodal to one cell over that induces that cell to make nodal, and then it passes it to its neighbor. And so each cell needs to have an intact nodal locus and be capable of making nodal for this to move one cell over at a time. And this is sort of a totally different mechanism from the way that people thought this nodal would work, which is that it gets secreted and then would diffuse and make gradients in that fashion. And so the question to us was, well, if that's what nodal is doing, what's the point of this extracellular inhibitor Lefty, right? Which gets secreted and is outside the cells, but if nodal is just getting past one cell at a time, is Lefty important? And so this is an in situ for where cells are making Lefty. So the nodal is in sort of purplish and the Lefty is in green. And it turned out that the Lefty is only made, as this nodal signal spreads inward, the Lefty is only made exactly at this front of signaling. So Lefty is a nodal target. It gets made in response to nodal, but only the newly signaling cells make it. And so that led us to think maybe this Lefty is sort of riding along at the front of this wave and slowing it down as it goes, and will change the speed at which the wave moves. And so to do that, people in the lab did this sort of very laborious feat of genetic engineering where they, we had these nodal cells that we modified. They knocked out both Lefty genes in these nodal cells so we could watch the signal propagate without Lefty. So here's sort of normal nodal signaling. It stays restricted to the edge. Here's when we remove Lefty, right? And so you can see this whole colony fill in bright with nodal, right? So in fact, you have this front of Lefty, and when you don't have Lefty, you get a much more rapid spread of the signal. And that has consequences for the fate pattern. So this is a marker of mesoderm. And when I remove Lefty, you can see this sort of altered phenotype where I have a lot more mesoderm than I otherwise would have. That's a consequence of this very rapidly spreading wave of nodal. So this also supports the idea that the timing of this nodal wave is very important. The Lefty, which is the inhibitor, sort of rides along at the front, governing the timing of this wave. And then when I remove it, the wave moves much faster, and I can differentiate. I get a much broader territory of this mesoderm gene. So we've done a lot with this system, a lot that I didn't have time to talk about, but this sort of summarizes where we are currently. And in the last few minutes, I'd like to tell you about sort of our very recent project where we've applied this system to at least one patient cell line and now to one gene on 8P with more in progress. And I should mention that this is all the work of a technician in the lab, Cecilia Guerra, who's on this call now as well, I think. Okay, so just to sort of summarize what the strategy of this would be. So you remember that I told you that you can reprogram cells to pluripotency. So that means that we can take cells from for example, 8P patients through skin biopsies or through blood draws. These can be then these reprogramming factors I mentioned. Typically, these 4 factors are induced in these cells. They're reprogrammed, and then they grow as stem cell lines, just like any other stem cell line in the lab, but they have the genetics of the patient. So they have these 8P modifications in them, and we can perform any kind of differentiation assays we would, just like we would with any other cell, and assay whether we get this same pattern or not. as a complementary approach to sort of narrow down the genes that might be involved in generating the phenotypes that we see here, we can also take regular induced, I call regular cells that make these sort of standard patterns and don't have known alterations to the genome. We can then alter the genome ourselves, one gene at a time, either knocking down the gene, removing one copy of the gene, removing 2 copies of the gene, overexpressing the gene, and seeing how that changes the phenotypes in these patterns. And comparing these two might tell us which genes are causative for the biology of phenotypes that we see from patients. And we're hoping that this is an inroads into seeing sort of, what is the biology of these patient cells and which genes can explain that biology. So I don't have a lot of data on this, but I have sort of one example of each of these. So this is the one 8P patient line we have to date. I'll show you on the next slide what its genotype is, but it actually has quite a drastic phenotype in this assay, right? So whereas normally we get rings of mesoderm, which might vary a bit depending on the exact protocol, here within the same protocol, you get this very expanded ring of mesoderm at the expense of this ectodermal center, right? So you see this has a big green center. The center is much reduced and you have much more mesoderm. And so we think this is, I should be clear that we don't know whether this phenotype that we can see in culture has anything to do with the actual phenotypes present in the patient. But we hope that assays like this are an inroads into the biology that might one day provide us with clues as to the biology that's happening in the patient. And this is an assay that we can work with, whereas before we had, no assays that we could work with. So we can go after the kind of genes that are involved in understanding these phenotypes. Okay, so that patient is identified by this label with the star here. It has both a deletion and a duplication on 8P. And as we looked at 8P, as developmental biologists, I know, we've heard some other talks about, bioinformatics, what genes might be interesting, but we identified just based on our sort of knowledge of development genes that were of interest to us because we know they're likely to impact the process of early development. So some of these are shown here. The TNKS gene, which is known as tankerase, is a very important gene in this Wnt signaling pathway, which is this spreading front of signaling, which is important for making the mesoderm. FGF signaling is another important mesoderm-inducing pathway, and there's both a ligand and a receptor on 8P. And there's also important transcription factors for the mesoderm, including GATA4 and SOX7. So we're interested in these genes. The first two we're targeting, as I'll talk about, are TNKS, and the FGF receptor is in progress as well. Looking at these genes together with patient phenotypes, we think can allow us to test different hypotheses. So one example is we've seen a mesoderm expansion phenotype in this patient sample. Based on the genes that are present here, we think it's possible that this results from the duplication of the FGFR1, so the receptor for FGF, which is a known mesoderm promoting pathway. And interestingly, there is a patient shown here, which has a fairly similar genotype, but a smaller duplication, right? So the prediction of that hypothesis would be that in this patient, we would see a less severe or non-existent mesoderm expanded phenotype if this gene is responsible for it. So this line is in progress. The other line that's in progress, I think, is this 504 line. These are being done by the foundation. And this is of interest to us, especially because it has a it's not a huge deletion, and it has, they're all big, but it's not, in the scheme of things, it's not huge. And it's around a couple of these mesoderm promoting genes, including this tankerase gene. And so we would predict, based on these genes at least, that this will have an opposite phenotype, that there will actually be a loss of mesoderm due to a loss of some of these mesoderm promoting signals and transcription factors. So this is, I think, my last slide. I'll just show you. So we the initial, as I mentioned, we've targeted this TNKS gene as well as the FGF receptor gene. So this means we've taken wild type cells, we've used CRISPR, we've isolated clones where this gene is mutated. FGF has proven a little bit more difficult, so that's still, we actually have clones now, but they haven't been tested, but these have been tested. So we have both complete knockouts as well as heterozygotes, which would more closely mimic what you would expect in a patient. And you can see that these do have the phenotype that we would have expected based on what I just told you, which is that this is involved in Wnt signaling, should be involved in producing the mesoderm. And you can see a sort of pretty strong reduction in the expression of this mesoderm marker in the knockout. There actually is a quantifiable reduction in its expression in the heterozygote, although, you know, as one might expect, not as strong as in the knockout. And so the key quantification is here. So the wild type is in black, the heterozygote is in blue, and the knockout is in pink. So, you know, this is not a very surprising result because the, you know, some of the functions of this gene were known, but we think it will be interesting to compare this with patient samples, which have this deletion, but also a bigger region. And it also serves as proof of principle that we can see phenotypes associated with deletion of single genes in this assay. And so we're hoping to do more genes and see different phenotypes and compare those with patients as well. So I'm going to stop there. Thank my lab, especially Cecilia, who did all the AP work to date, and our funding sources, you know, particularly Project AP, who is now funding this work on patient samples and genes on AP that we're doing. And I'm happy to take any questions.
Thank you so much. That was really amazing. Lots of background, lots of detail, but super important setup. And then I think this very tantalizing set of preliminary results, and then some results that are hopefully going to start to unfold over the next few weeks and months. But I think, Faye, you raise your hand first. Anyone? We've got about 10 minutes here, and then we hit the 60-minute mark. So I want to be respectful of everybody's time. This has been recorded. So either raise your hand if you want to ask your question live, or drop your question in the chat. And if you are asking your question live, please turn your camera back on. So Faye, please kick things off.
I actually was just clapping, but I do have a quick question. So that was a brilliant talk, Aryeh. I really enjoyed it. Thank you very much. I'm just wondering how much variation you see in those zones with different controls. Is it always like boom, you know, like within 1% variation, or is it like 20% variation just in normal IPS cells?
Yeah, that's a great question. So I should, if I go back here, I should distinguish between sort of colony to colony variation in the same experiment, and then variations between experiments. So if the experiment is done well, the colonies are very reproducible. And so, you know, you get a sense of that from these error bars here, right? These are, I think we did 7, you know, we imaged and quantified 7 colonies of each of these genotypes in this experiment, and the variation is quite small. So this change is quite reproducible. Experiment to experiment is trickier because there are variables that influence this process. So one sort of very obvious one is the density of cells. So exactly where the sort of qualitative result is extremely robust, right? You always get the same order of things, you know, the same fates in the same order, but you know, whether this, say, outer layer is, you know, 2 cells thick or 3 cells thick, right, that will depend on things like cell density. It will depend on the dosage of the recombinant protein that we add. And we found actually that if one is being very careful, these, you know, if one is not looking too carefully, they're fine in the freezer for months, even years. But we found that, you know, if you really quantify it, you know, something 2 years old will give you a slightly narrower band than something new. So that kind of variability, definitely, you know, I would say it's not enormous, but, you know, whereas this would probably be in the realm of, you know, 5%, you're talking more like 25% variability. But that, what that means is it's very important for us to always do the controls in the same experiment. And in particular, you know, we need to be very careful. You know, experiments where we seed the same cells and treat them differently are somewhat easier than these experiments where the cells have different genotypes, because we have to seed them separately. We have to be very careful to keep their densities consistent and things like that. And also it means that sometimes there may be sort of primary causes, there may be sort of more proximal causes to these shifts that don't really have to do with the patterning. So for example, if there are cell cycle differences, even if you seed the same number of cells, if some cells grow more than others, then you may see a difference in pattern, and that's something we need to be careful about. So we're, cell density is something we're always looking at, but and that's the one that's hardest to control sort of between cells of different genotypes here. But at least within this experiment, which is being repeated, if you look at the overall sort of density in these colonies, it's similar enough that we feel pretty good about this result right now.
Thank you.
Like Nicoletta, you have your hand raised.
Hello, thank you very much for your talk. It was great. I just also had a couple of questions related to your experimental points that you have just mentioned about the cell density. Because there are cells that are still dividing in there, are you likely to have these colonies as a flat surface colony, or are they likely to end up going in three-dimensional a little bit?
Yeah, so that's a very good question, and that's something we've looked at pretty carefully. So what they start out as, so the epiblast is an epithelium. So these colonies start out as a single layered epithelium. And what happens is, particularly in these mesendoderm fates, which are made in the primitive streak, that differentiation is associated with an EMT transition, so an epithelial to mesenchymal transition, where they leave this epithelial layer, and they actually undergo this transition in this sort of blue ring here, and then they crawl under the colony. And so, as time goes on, you get a multi-layered colony with but in sort of an ordered way, with mesendoderm underneath and the epiblast or ectoderm on top. And we've seen, there's sort of different ways we do this assay to model different things, but and there can be very three-dimensional aspects to it, but they're also, at least for the first few days before things get really overgrown, the morphogenesis as well as the fates are quite reproducible. Cells are sort of programmed to do very particular things.
And at which point are you doing your genetic manipulation? When they are before plating for colonies, or?
Yeah, so these are established. So we will take the iPS cells, we'll do the genetic modification, we'll establish clonal cell lines, we'll genotype those clones, and so then that's an established line, and then this assay takes 2 days. So the assay will be done with that established cell line that's been genotyped. So we're not, we're not sort of, some people who do screens, you might, if you can get a high enough percentage, you might just sort of nucleoffect the cells and hope it's high enough to see a phenotype. These, at least for these genes where we're not doing that many, we're sort of being quite rigorous about the genetics and getting clonal lines that we know the genotype of them.
And are you nucleoaffecting or using lentiviruses?
We're nucleoaffected. So they're nucleoaffected. This is all done with CRISPR. So you're nucleoaffecting. Actually, what we do now is we don't nucleoaffect any DNA. We're nucleoaffecting the Cas9 protein together with the guide RNA complexes, which you can buy. And that's actually, we've gone over to that recently because it's tremendously more efficient than using plasmid. We can sometimes get 80–90% modification of the genome. So.
Thank you. Thank you.
I had a question. So if your hypothesis about FGFR1 duplication being sort of driving the mesoderm expansion, does that have any therapeutic implications, or would you have to basically apply an FGFR inhibitor at like day 17? And how would you even know? Like, is there any therapeutic implication, or is this more just like, okay, you can now start to show the mechanism of the neurodevelopmental defect, but I'm just trying to get at, could any of this be happening late enough in development where you could first detect it and then actually maybe even do something about it in utero?
Yeah, so I mean, I think that's a great question, and I think we don't know the answer. I think our hope is that, the biology that is in early development is it's not going to be the same, but it's going to involved, FGF is involved in many different stages of development. So if you have a mesoderm overabundance, by a certain point, it may be too late to correct that, but there are still going to be consequences of elevated FGF signaling, assuming that's still ongoing. And we don't know. There may even, FGF plays obviously lots of roles in, postnatally as well. And so we couldn't prove this in any way with our systems, but a hypothesis would be that, having I would say a low dose of not to inhibit the pathway, but to reduce the levels of the pathway, is something that, I think what you would want to do is look at different stages, measure FGF signaling through things like phospho-ERK and compared to controls. Are you seeing elevated FGF signaling not only early in development, but at other time points, and can we associate that with phenotypes that we're interested in reversing? So I think for me right now, this is, we're interested in understanding mechanisms of this early stage of development, but this also provides sort of clues as to the general aspects of biology, which may be important later, right? All these pathways are used recurrently, both in development and postnatally for all kinds of things.
Well, I think that's a major take-home message here. We've got about 1 minute left. I just wanted to call out anybody who has a question or comment here as we wrap up. If not, then I really want to thank Professor Warmflash for a really tour de force here, masterclass in early neural development and how you can control it in a lab, create these basically embryo proxies that you could process in 2-day turnaround time. And as you said, may not be able to intervene at some early day in neurodevelopment in an early embryo, but these pathways also are chronically either overactivated or underactivated, whether it's deletion or duplication. So I think there's always going to be implications beyond neurodevelopment, even though that's how we've started out kind of the project sponsored work. Okay, great. With that, then I want to wrap things up. And again, Bina, any remarks here to close us out? Otherwise, I'll just say thank you everybody for your time. This will be available by video link. Whitney will make sure that is circulated. And if you have any other follow-up questions, please reach out to Whitney or me or Travis or anyone else on the Polaris side. We can connect you with Professor Warmflash and get any questions answered here. But I thought this was a great session. And again, thank you, Aryeh, for your time.
Okay, yeah, thank you all.
Cheers. Have a great rest of your day, everybody. Have a great rest of your week.